and welcome to this webinar on sample size allocation for multi-regional clinical trials, which is the latest in our monthly series of webinars. In this webinar, we will talk about how larger global clinical trials are trending, why these multi-regional clinical trials are important, as well as some of the current regulatory guidance in the area. We will also discuss various design considerations, including sample size allocation for the various regions in the trial. And we'll go through some worked examples of those calculations in nQuery as well. So just first and foremost, before we get started with today, I'm just going to run through some housekeeping first. So as always, today's webinar is being recorded, and that recording, along with the slides used and the example files, will be sent out to you shortly after we conclude here today. Just one point to note this time, however, is that the tables we're going to be looking at today are part of an upcoming 9.6 update. So we'll be looking at a preview in a development version of nQuery, and so if you want to play around with the example files, you'll need to wait until the update 9.6 comes out in order to have a look at those files yourself. If you do have any other questions, please feel free to type those in the chat box. I'll do my best to leave some time for questions at the end, but as always, anything I don't get to answer during the webinar, I'll make sure to reply to you in a timely manner as well. And that goes the same if you have any questions after the webinar. Of course, you can email those in, and I'll get back to those as well by email. You can also use this chat box to inform us if you're experiencing any technical difficulties, and one of my colleagues will do my best to resolve them for you as well. Okay, so before we go any further, just a quick introduction. My name is Denis Desmond, and I'm a research statistician here at StatSols, and I am going to be your host for this webinar that's brought to you by nQuery. Of course, as well, thank you all for taking the time to join us today for our webinar, and we'll have a quick look at the agenda. So I already mentioned a couple of the things that we're going to cover, but just a very quick rundown. We'll start off with an introduction to the topic and then give an overview of multi-regional clinical trials. We'll then look at some design considerations for multi-regional clinical trials before talking about sample size determination, and at that point we'll look at some worked examples in nQuery as well. Finally, we'll wrap things up with discussion, conclusions, and of course questions as well, if we have sufficient time. Okay, so as I mentioned, this webinar is brought to you by nQuery, the complete solution for optimizing your clinical trial designs. nQuery is very widely used in pharmaceutical and biotechnology industries and offers sample size determination and power calculations from preclinical research through to post-marketing studies, with the main focus being on confirmatory phase 3 trials. nQuery features over 1,000 scenarios for fixed-term, Bayesian, and adaptive studies. And if you're interested in learning more, you can see the resources section of our website for demos, worked examples, previous webinars, and of course, you'll be able to find out about future webinars from there as well. So nQuery operates on a tiered licensing model, and so each of the 4 tiers gives you access to a different feature set depending on the tier you purchase. And so it's obviously a progressive model, so if you buy or upgrade to higher tier, you still have access to all the features you had in the previous tier as well. And so if you'd like any more detail on this, you can follow the URL here at the bottom of the page once the slides have been sent out, and this will take you to the comparison tool page, which includes a detailed breakdown of the tier required for each table that's offered in nQuery. On that note as well, if you don't already have a license or you'd like to try a higher tier of the software, you can get a free trial of the full features of nQuery from our website, and I'll go through that more in the end and provide as well the contact details if you'd like to get in touch with us to get any further information. So that's the kind of introductory housekeeping items covered. So we can get started on the first section. And as I said, that's an overview of multi-regional clinical trials. So first and foremost, a multi-regional clinical trial is defined as a trial that is conducted in more than one region under a single trial protocol. In the guidance, region is the general term that can be used to refer to a geographic region, a country, or a regulatory region. And so a regulatory region would be something like the EU, where there's a group of countries that have a common set of regulatory requirements for drug approval. Traditionally, however, drugs are conducted in a single region in collaboration with the relevant regulator for that region, with the goal of receiving approval for that new treatment in that region. And then if they wish to make this drug available in additional markets, they can approach other regulators, and they may request a bridging study if not all the regulatory requirements were met in the initial study, or that they feel that the study population wasn't fully representative for this patients in their region. And so that could be because of a number of factors, such as difference in genetics, culture, medical practices, just as a few examples, but that's something that we're going to talk about more in a second. And as well, sorry, by bridging study, again, we'll talk more about that in a second, but a bridging study is one that's performed in the new region to provide supplementary pharmacodynamic or clinical data to allow the extrapolation of foreign data to the population in that new region. And so if a bridging study can show that the safety and efficacy profiles in the new region are similar, then the pharmacodynamic and the pharmacokinetic data will be sufficient to bridge the foreign data. But if it seems like that won't be enough, then it may need another confirmatory clinical trial to be the bridging study. And so as drug development continues to become more of a global venture, and also because of the potential efficiencies that these multi-regional clinical trials can offer, they've been getting an increasing amount of attention of late, and that's what we're going to talk about there now. So why are these multi-regional clinical trials important? Well, firstly, from a sponsor point of view, a well-planned multi-regional clinical trial can potentially avoid the duplication of studies that sometimes might be required by the bridging study. And if the plan is to expand into other markets, then a multi-regional clinical trial can be a more efficient use of limited time and resources, because obviously drug development is a very lengthy process, very expensive process, and so well-planned multi-regional clinical trials can help streamline the use of these resources. And so if you look over here on the right-hand side, we have a diagram kind of breaking down the two different strategies. And so in the first case, if you see the kind of independent strategy for local trials in region A, the single kind of development pathline starts off, and then once it becomes clear that additional information will be required to bridge to the new region, say, the B region, you can see that there's this staggered approach and delayed start. And so, from the point of initial submission here to final regulatory approval, you can see where there's this large delay in region B. There's also then because this might not necessarily have been fully planned. There's the requirement of trying to bring sites online in region B, and the various logistics that go with setting up a clinical trial. Whereas on the other hand, in the global strategy, the single protocol is discussed with all the relevant regulators from the outset, and all of the planning is done, and the different design considerations that we're going to talk about in a while have been considered. And so with this strategy, it allows for simultaneous submission for approval in the various regions, and the delay you can see here has been removed by doing so. And so obviously that has a patient benefit as well, because while from the sponsor point of view, this is obviously a delay in getting a product to market, but from a patient point of view, that is a time lag where a potential gold standard treatment is not available in their region. And then another benefit for the multi-regional clinical trials for patients is that in the cases of extremely rare diseases or conditions where enrollment is always expected to be slow or extremely slow, then it might be difficult to have a large confirmatory trial consisting of patients from one particular region exclusively. And so an appropriate multi-regional clinical trial will be able to reduce the timeline required for a trial like this, or also include regions that may not have otherwise been included, in that particular scenario. So, in the past, when drug development was more concentrated in fewer places, like the US and Europe, for example, bridging studies and the acceptance of submissions based on foreign data were more of a concern for some regulators than others. However, now the industry is becoming so much more global, and drug development is a worldwide effort, and as a result, bridging studies and multi-regional clinical trials are of greater interest to all of the regulators as they look to ensure that they are sufficiently represented in these global trials. And so, on that note, we're going to just click on here to our second section, which is multi-regional clinical trial design considerations, and we're just going to have a quick talk about the regulatory context. So the first major guidance on the acceptability of foreign data was the ICH E5, which was issued in 1998. And the scope of this document was to try and minimize the need for duplication of clinical trial data by, outlining and recommending strategies that can help with the acceptance of foreign clinical trial data, and when there may be potential reasons for populations to differ. And so the first part, the kind of low-hanging fruit was to just make sure that in the original study that you'd satisfied all regulatory requirements that could be asked of you in the new region. Otherwise, it goes without saying that you would need a new trial in order to achieve approval in the new region. But then, obviously, the more difficult part is the extrapolation of this clinical data from the foreign patients onto the new region. And so, if there's a belief that there's some factors that could alter the safety and efficacy in the new population, then a bridging study would be required. And so ICH E5 outlined that there's kind of two groups for ethnic factors. So there's intrinsic factors, which are related to genetic and physiological differences. And then there's extrinsic factors. So those are related to like cultural differences, environmental differences, differences in medical practices in the different regions. And so obviously in this document, they kind of highlight some potential differences, and as well also they kind of mentioned that it's possible that a drug may or may not be sensitive to either the intrinsic or the extrinsic factors, or both. And so in the appendices of the guidance, there's a section outlining properties that don't say for certain, obviously, which drugs may or may not be sensitive to these factors, but factors which have been predictive in the past for the sensitivity of the drug. So just one such example is that if the drug has a flat concentration curve for efficacy and safety in the recommended dose region, then it suggests that the drug is well tolerated, and so then it is less likely to be sensitive to ethnic factors. However, the reverse then is also true. If the curve is steep in the dose region, then it suggests that that the drug will likely be sensitive to ethnic factors. And so this was obviously a very important first piece of guidance, but it still kind of didn't cover all of the scenarios for maybe moving towards a global trial rather than just a bridging study. And so following ICH E5, during the working group Q&A, the Japanese pharmaceutical and medical device agencies, PMDA, were key proponents for this idea of global trials, and like this shift away from sequential drug development to this simultaneous global model. And so then, as a response to this suggestion during the working group Q&A, their Ministry for Health, Labour and Welfare issued a 2007 guidance, basic principles for multi-regional clinical trials. And so again, this was done to, first of all, encourage sponsors to embrace the idea of these global trials and encourage Japanese participation in them. So it kind of laid out in a question and answer format certain things that regulators in Japan would require in order to carry out one of these multi-regional clinical trials, and also it specified two criteria for determining the Japanese representation in these large clinical trials. And so that's something that we're going to talk about more later. But just another point to note about this document is that it was highlighted in it that the reason that they were so in favor of this simultaneous drug development was because of the severe drug lag that was being faced by Japanese patients. And so drug lag is the terminology that they gave to that kind of problem with sequential drug development that I mentioned earlier, where in this case Japanese patients were receiving treatments years and years after other markets, and so in order to try and bring that wait time down for their patients and improve overall public health, the Japanese regulators wanted to kind of push forward this idea of sequential development. or sorry, move away from the idea of sequential development to this simultaneous global trial. And so, of course, naturally, I should have said it a moment ago as well, like this isn't an exhaustive history of all the different regulators' guidance. That would use up all of our time today and not make for the best webinar. There were many others, China and South Korea especially, that also played a part in the move towards these larger global trials. These are just some kind of milestones to give context, set the scene for kind of the few talking points that we want to hit on today. That's just something to point out there, sorry. Then the next guidance that I want to mention was ICHE 17, issued in 2017. And so this was focusing on like the kind of framework for conducting multi-regional clinical trials, with the specific goal of receiving simultaneous approval from multiple agencies. And so we'll, talk about that kind of, what needs to be considered in a global trial on the next slide. But, as I mentioned earlier, the nature of the drug development market now is that it's continuing to grow. It's becoming more global. And so these global trials are becoming more and more popular, and it's something that more regulators are having to consider. And so this regulation is going to continue to evolve. For example, just last year, the FDA released their first draft guidance on oncology multi-regional clinical trials. And, in that document, they highlighted, that they wish to, first of all, aid with, the kind of number of sponsor queries they were getting regarding multi-regional clinical trials, but also to encourage the use of these global trials, to facilitate the inclusion of US patients, something similar to what Japan's facing, because, in the document, they've noted that overall, the number of US patients is decreasing in these oncology trials. So, as I said, this list isn't exhaustive, and, it's going to be continuously updated as this, becomes a more and more global endeavor. But for now, yeah, let's move on to some of the design considerations. So, obviously, like any other clinical trial, there are many important considerations that have to be made at the planning stage. But naturally, given the scale and the involvement of multiple regulators, there are some unique points as well that you have to think about when conducting a multi-regional clinical trial. Excuse me, sorry. So on the table here, we just have kind of broken it into categories, some of the considerations and just some examples of them. We can start off here with clinical, and, we've spoken about the importance of accounting for the various intrinsic and extrinsic population and ethnic differences. But, when you're conducting the multi-regional clinical trial with the single protocol, and you have differing medical practices, different definition of the disease criteria, these things need to be considered, and what is this true in one region might not necessarily be true in another region. So there needs to be careful planning at the outset as to how these things are going to be defined. There's also ethical decisions to be made, such as the choice of control group, and then different regions might have different standard treatments, and the gold standard might not be available in all of the regions, and different regions might have different perspectives on placebo versus active control. And so these are all things that need to be taken into account. In terms of statistical considerations, there obviously needs to be a definition for consistency within the regions, and a determination of how to allocate the overall sample size among the regions, but also to determine that overall sample size based on the anticipated effect. The regions themselves need to be pre-specified. So we mentioned earlier that regions can be geographic countries or regulatory regions, but also if they are predefined, then certain regions might be pooled on the basis of kind of these intrinsic or extrinsic similarities for the basis of analysis. And so again, if that's the case, that all needs to be predefined. And as well, there needs to be a pre-specified strategy for accounting for the multiplicity of testing and controlling the type 1 error. Then from a regulatory context, obviously it goes without saying, but early communication with the regulators is always a good first step. Just to again clear up any requirements that will be will have to be met, in each of the regions, and it will also make it clear what initial documents and data need to be submitted prior to actually starting the multi-regional clinical trial. So just as an example, in that basic principles document that was issued by the Japanese Ministry for Health, it outlines that ahead of time, the planned dosages need to be shown not to cause any safety problems for Japanese patients. So they will obviously accept if there's an appropriate way to extrapolate safety data to the Japanese population. But if that isn't deemed sufficient, then there needs to be a phase 1 study for the Japanese population. So again, knowing things like that ahead of time by getting in contact early with the regulators will help at the planning stage. Another important issue with the regulator is the guidelines around the determination of enrollment. In the case where initial enrollment is completed, is the strategy to continue enrolling or does enrollment stop? But then also, if there's going to be interim looks, will sample size be terminated at that point based on futility or efficacy, things like that. So then, obviously, operational as well. Many considerations here, given the fact that this is a trial on a much larger scale with many countries involved, and so there needs to be a consideration on the appropriate number of sites and balancing the recruitment across those sites, as well as ensuring that there's standard equipment and that the personnel have the sufficient training at each of the sites to ensure that it is the same across all the regions. Again, because of the operation under a single protocol, there needs to be consistency across the sites during the trial in terms of things like the data that is collected, how it's recorded and stored. Is it going to be paper or electronic? Things like that. There's also like supply chain issues. so again, this is a trial that's happening on a global level, and so the test drug needs to be shipped to the various sites at the appropriate times, along with any comparator treatments. And there needs to be facilities there to store it correctly. There needs to be decisions about whether there's going to be individual distribution points in the different regions, or if it's going to be centrally managed. So there's just all of these concerns that need to be addressed in the planning stage. And then lastly, there's the commercial considerations. So the first one is to kind of assess if the new treatment is going to be of value to patients in the various regions. What is considered to be the gold standard in treatment can vary from region to region. And so, of course, there's potential for patient benefit there. And then the other thing to consider is if the goal would be to expand the market for this product if it had been developed in a more traditional way, because obviously if there's expected to be a limited market for the product, or very different effect in the presence of these ethnic factors, then it might not be a suitable candidate for a multi-regional clinical trial. So there's kind of a list of things here. Again, like I said, not exhaustive, but just to give you an idea of the various considerations that need to be taken into account. But one that we mentioned, and one that is of importance for all clinical or multi-regional clinical trials, is the allocation of the sample size to the various region. And so, in ICHG 17, they state that there's no universally optimal approach to allocation, as it depends on so many factors like the size of the region, the impact of the ethnic factors, and the prevalence of disease. And so as a result of that, many regulators will have their own guidance as to how they feel the sample size should be allocated out. But the Japanese Ministry for Health, Labour and Welfare, in that 2007 document, kind of issued the first formal criteria. And so, it's something that's been widely discussed in the literature relating to multi-regional clinical trials. And so that's what we're going to focus in on today, because, in other cases, it's more of a strategy rather than having a formal criteria. And so we'll take a look at this here. And so the two main goals, like I mentioned, of the clinical trial on a global scale is to first demonstrate the treatment effect in the overall population. But then there is that secondary goal to assess the consistency between the regions. And so that's what's defined here, and that's what is missing from a lot of the other guidances in terms of like a formal guidance for that definition. And so here, consistency is represented by 1 minus beta dash, and values of 80% or higher are recommended in the guidance for this value of consistency. And so in method 1, it's concerned with the proportion in this case, the Japanese population, but it's obviously can be generalized to any region that is part of an overall larger trial. And so what the method 1 is saying is that the proportion of the treatment effect in the Japanese population relative to the overall treatment effect needs to be greater than this value pi with a probability of 80% or higher. And so in the guidance, pi is set at a minimum of 0.5. But then method 2 is concerned with there being an overall treatment effect in all of the regions. So in this case, it's the probability that each region has a positive effect is greater than the consistency threshold in this particular case. And so the guidance put forward these two methods but didn't include, sorry, I should say, sample size calculations. And so there's been many papers discussing this, and so that's going to segue us into our next section, which is on sample size determination. And I'll pick back up with the PMDA methods there in a second, but just obviously we've had a lot of talk about regulations and things like that. So what we'll do is we'll actually start this section with just a worked example for a sample size calculation in a bridging study. So this here is just a hypothetical scenario that's based on calculation data from table 1 of the paper here, sample size requirements for the evaluation of bridging studies by Liu et al. And so this table is just a number of calculations for different scenarios, but I've just pulled one out here for us to use as our example today. So we're imagining that there was a clinical trial conducted in region A to establish the safety and efficacy of the new drug, and the mean difference was 2 and the common coefficient of variation was 0.4, and then a sample size of 1,000 was needed to meet the target power of 80%. So then if the sponsor wanted to, or was required to carry out a bridging study in the second region, then sample size of 160 would be required in the new region to achieve 80% power target, given the equivalence limits of minus 0.2 to 0.2. So that's summarized here on the right-hand side. And we will switch over to nQuery. And we have the table for bridging studies, equivalence test for bridging studies for two means. Open here. And so I mentioned at the start of this webinar that this is a development build of nQuery. And so this is a preview of an upcoming table in our new release. So just as a quick reminder, you will receive the example files later today, but just until you have updated to 9.6, you won't be able to open these right away. And then just quickly, for those of you that might not be as familiar with nQuery, this is how most of the tables look. On the left-hand side here, we have various row names. So they indicate the input required. Each column will serve as a separate calculation. These yellow highlighted rows are values that can be solved for. Calculation can automatically run if you fill in the appropriate number of columns, or you can select from this drop down here which solver you'd like to run. The gray values here are read-only rows, and you don't need to provide an input for them, but they will provide additional information once the calculation has been completed. And then over here on the right-hand side, you have the help cards that just give additional information on each of the inputs required. So that's the basics anyway, and we can start off with our example and fill in our values here. So 0.05, 0.2, our mean difference 2, and original study sample size is 1,000. Common coefficient of variation was 0.4, and we want equal allocation in our bridging study sample size. And so then if we go down here and we target 80%, and so we can get 159. And so obviously that's the minimum sample size required to achieve the target power. And if we wanted equal allocation again, we could put 80 patients in each and would of course achieve the power as well. So now just as a quick sneak peek of another feature that's coming in our upcoming release. Those of you familiar with nQuery will know we've got two options for plotting up here. One will plot power versus sample size, and the other will allow users to plot selected rows in a graph. And so this is the familiar dialog box that you'll have seen before. And so it allows you to specify your X and Y axis. And so I'm going to set up one here where we look at the original study sample size versus the bridging study sample size. But the little preview that I want to give is a very exciting new feature that we're adding in 9.6, which is custom X-axis values. And so I just should quickly say previously, in case people weren't as familiar, you could specify a minimum and a maximum value for X, and then a step size, and you get equally spaced points along the graph depending on your different inputs here. But with this new update, we've added the ability to input custom X-axis values, and so by default it's populated here with the values from that we'll say default method for implementing a chart, but we can clear all those and you can enter in then whatever values you wish, separated by space up here in this input field. So just say, for example, that I wanted to compare the other values in the table that I mentioned. We could fill out quickly here just these and add those in. And from this point you can change the number of rows that you'd like to input. You can obviously clear all and start again. You can delete individual rows, or you can go back up here to add in more. But like I said, just for the purposes of the demo, I'll just run this like it is. And so you can see here that we get the plot with our own custom points. Now, obviously, it's not a very smooth curve because it's only 4 points, but it's, just for the purpose of demonstration. And so this is a brilliant new feature. And, again, as I said, that will be coming shortly with our 9.6 update. And as I said, it's obviously not a very smooth curve, but you can see from it the trend that, like, as the original study sample size increases, then the bridging study sample size that's required will decrease then as well. Okay, so we'll pop back to the slides here for a second, and we can talk more about those PMDA methods. So just I'm conscious of time here, so I won't spend too long on this at the minute. We'll talk more about it when we're kind of looking at the examples in nQuery. But I'll just, quickly just touch on it, as it highlights again the two objectives of the multi-regional clinical trial. Again, to show an overall effect and then to show consistent effect. And so in method one, which is based on a paper, this version of method one is based on a paper by Kwan et al. From 2009. You can see that in the overall sample size calculation, it's pretty standard. It doesn't depend on any consistency definition. It's just the effect size, the significance level, and the power. And the same is true in the overall sample size calculation for method two, based on this paper by Kwan et al. From 2008. And so then it's the definition of consistency, and then the region subsample that will impact that. And so here again, 1 minus beta dash is for the consistency, but here it's defined by this capital P. And you can see in this that this is where the sample size and the effect in the new region is impactful in the sample size. And here I should highlight that the lowercase pj here is the proportion of subjects in the jth group, or jth region, I should say, sorry. And so, yeah, like I said, we'll talk more about it in the examples, but we'll just go back to nQuery and talk about it more there. So our second work example today comes from that aforementioned Kwan et al. Paper, and they describe a scenario for a multi-regional clinical trial designed to evaluate drugs' effect on baseline HbA1c, which is a measure of average blood sugar. And this trial used an unbalanced design and was overpowered to detect a difference in HbA1c of 0.5%. And so with 372 patients in the treatment arm and 186 patients in placebo group, the study had an overall power of 99%, assuming a common standard deviation of 1.3% and two-sided test at the 5% level of significance. And so for pi equal to 0.5 and consistency equal to 0.8 or 80%, then the sample size for the Japanese patients were 26 in the control group and 51 in the treatment group, or 52 to satisfy the 2-to-1 ratio. So again, we have that summarized here, and just one thing to add is that it's assumed that the treatment effect is the same in both regions, so the regional effect size ratio will be set to 1. So if we hop back over to nQuery now, and we open up MT43, which is our sample size for the PMDA method 1, and we can start filling in our values. And so, as I mentioned there, we're going to fill this as 1, but I'm going to just talk about it again more in a second. The testing threshold was 0.5, and the sample size ratio was 2-to-1. The consistency target was 80%, and its power was 99. Okay, so the first thing to note here, obviously, is the kind of warning here. So if you look down in the output statement, the achieved power was 99.02%. And so oftentimes going above 99% is beyond the realistic range for a model or the assumptions, the formula being used. And in this example, as I mentioned, it was deliberately overpowered, so it's something that they were aware of. But nQuery, once the power goes above 99%, just because of the fact that it sometimes can be on the realistic range, nQuery is going to flag that for you. If we look at the rest of the results, then you can see that in order to meet both of these targets, approximately 13.8% of the population or the patients in the overall study will need to be from the Japanese population. And then you can see down here we have the overall sample size as well as the regional sample size, and again with that 52 and 26 split for the 2-to-1 ratio, as they mentioned. So the other thing to talk about is this regional effect size ratio here. And so obviously this is a value that should always be very close to 1. If it's not 1, by definition, the criteria is to demonstrate consistency across the regions. And so obviously this will allow for, slight variation if you expect that the effect might not be exactly equal, but expecting large differences, makes it very difficult to demonstrate consistency. And obviously maybe that particular trial won't be a candidate for, or that particular treatment won't be a candidate for multi-regional clinical trial. But just to demonstrate, if I just copy that initial calculation into 2 further columns here, and I just tweak this ever so slightly, you can see that we still will meet the targets for consistency and power, but we're going to need to have a higher proportion of the sample size come from Japan or whatever the region of interest is, in order to ensure that we can meet that consistency target. On the other hand, then if we bring it slightly above 1, you can again see that we have hit the 2 targets here, but that the population required is reduced, because again, in order to show consistency, the population in Japan needs to be slightly lower when there's a higher effect size. And so that's kind of intuitively what you'd expect as well. And so we'll go back now just to the slides, because again, just conscious of the time, and I want to get through our last example here. So this is again from the Kawai et al. Paper mentioned earlier. And again, this is a hypothetical example scenario created from data that was in a table in that paper. And so, imagine that the sample size was to be split 25%, 25%, and 50% among each of the 3 regions. Then an overall sample size of 1006 is going to be required to achieve target power of 80% and a consistency of 80%. I'm assuming overall effect size of 0.125 and common standard deviation of 1. And again, summarized here, and we'll just jump back over to nQuery. And so if I open up MT44 here, which is our table for method 2, I'll start filling out the values, and you'll see here once I put in the number of regions, this additional table down below opens up. And so in nQuery, for those of you that aren't familiar, side tables are used as a way to provide more information to a particular cell in the main table. And so sometimes these are alternative ways to calculate a certain value, or in this case it'll provide a breakdown of the sample size for the different regions. So now I'll just fill that in there first before I go back up to the main table here, and we can fill out the rest of our values. And you can see here that we hit the thresholds for power and for consistency. So I'll just go back to the slides for a second and just go back up here just to reiterate something that I said a second ago. So, the overall sample size and power, won't be impacted by the proportion of the subjects in each of the group. It's this value for consistency here that will change depending on the allocation between the regions. So if we go back to nQuery and we just copy this over here for a second. So in Quietall, they talk about how there's the maximum consistency will be achieved when there's equal allocation across all the regions, and I think intuitively that makes sense. And so you can see here that when we equally split the sample size out across the regions, that excuse me, sorry, we achieve approximately 85% consistency, and so that's the maximum value for this given set of inputs. But so then a sort of corollary to that is that there's a minimum allocation that each group needs to have in order to achieve the target consistency for a set of values. So if I just copy this over here again, and instead I put in, say, 20% in each of the first two groups, then you'll see that approximately 78.9% consistency. And so in the paper, they've shown that for this particular set of inputs, it's roughly 21% is the minimum sample size requirement in our minimum allocation for each region in order to achieve the target of 80%. Okay, so that was our last example there now. And I am just obviously conscious that starting to be tight on time, but just very quickly, those were two methods obviously proposed by the FDA, but that's obviously not the only way to allocate sample size. And so here we have a list from ICH E17, and they've noted that these are just strategies that have been used in the past. It's not necessarily, the only way to do things, or it's not even an exhaustive list, and that, innovation and further development is still encouraged. their recommendation in the ICH E17 guidelines is a balance of these first two strategies. And so that way you kind of blend the two of those to get the advantages of both. So the first thing is, like with proportional allocation, you allocate the patients depending on the size of the region and or the prevalence of the condition. And so, like, one advantage of this is that, obviously it can allow for quicker recruitment, but the downside then is that it could end up with one region dominating the trial, and some regions may not have the chance to recruit sufficient patients to allow for those consistency assessments to be made. And then equal allocation, as we mentioned a minute ago, will maximize the power, but then by maximize the power to show consistency, or I should say maximize the consistency. But then depending on the sizes of the regions and the prevalence of diseases, it may mean that recruitment for one place is much slower compared to the others. And so, as I said, they recommend blending those strategies. So, perhaps starting off with equal allocation and then adjust based on prevalence data to ensure that there's sufficient overall sample size, while also having the necessary regional allocations to demonstrate consistency. And so then just some of the others quickly. Allocation to determine or to demonstrate, a pre-specified portion of the overall treatment effect in a specific region is impractical if you end up needing that requirement for multiple regions. And likewise, allocating sufficient subjects to show significant effects in each region is going to inflate the sample size and kind of moves away from the core principle of a multi-regional clinical trial. And then the last option is a fixed minimum number of patients. And so obviously this can be useful if there's a valid reasoning behind the choice of the minimum value, in order to assure sufficient sample size and things like that. But otherwise, it's just going to lead to inefficient recruitment or inflated sample sizes by requiring a fixed number if that number isn't picked with the right reasoning behind it. So yeah, sorry now that was a bit rushed there towards the end. I realize I've just gone over time there now, so we'll quickly wrap up there with the discussion and conclusions. Just a reminder, we began discussing the importance of considering intrinsic and extrinsic differences in populations as described in ICH E5 when you want to offer new drugs into other markets. We then spoke about how traditionally drugs will be developed for one market in conjunction with that single regulator, and then bridging studies may or may not have been used on an ad hoc basis if required by the regulators when product was to be expanded into a new market. And then we spoke about how the globalization of the drug development industry and the desire from various regulators to minimize the drug lag for their respective patients and the inclusion of an appropriate sample from their population in these global trials has led to the embracing of multi-regional clinical trials and the benefits that they can bring. And then lastly, we spoke about how there was no undisputed best way to design a clinical trial, and how the allocation of subjects in a multi-regional clinical trial would depend on many factors, like the disease prevalence and the various regulations that are in place in the different regions. But we did speak about some potential options, and we looked at sample size based on the PMDA method 1 and method 2, which is, as I said, frequently discussed in the literature. So yeah, obviously I'm sorry that I've gone over. I will get to the questions as soon as I can. I will reply by email. I'm very sorry again for that. As I mentioned at the beginning of the webinar, if you don't already have a license for nQuery and you'd like to try it out, or you'd like to try the features from a higher tier, you can go to statsols.com/nQuery-demo and get a free 14-day trial with no download or install required, just access through your browser, no credit card requirements, just need to provide an email address. This trial will give you access to the latest version of the software, all of the tiers, base, plus pro and expert, so you'll be able to try out all the different tables for the fixed term, adaptive designs, and things like that. Just a quick reminder, today, featured a development build of nQuery, so those tables won't be available in the trial until the release goes out, and I'll mention it again in a second, but same goes for the example files. Those you won't be able to edit those or have a look at those until the update comes out. So with that, if you do have any other questions, you can email them to info@statsols.com. Myself and one of my colleagues will get back to you. Feel free to reach out if you have any feedback, if there's anything you'd like more detail on, or if you have a suggestion for a topic in the future webinar. We'd be delighted to hear from you. There's also more information and our full library of past webinars available on our website, statsols.com. And then at the end of the slides as well, you'll receive these shortly. You can see that there's the references as well. You'll also get the recording and the example files, but as I mentioned, those tables are part of our December update, and so they'll be available to you shortly. But again, if you have any questions with any of that, you can get in touch. So with that, I know I've gone over time, so sorry for that. Thank you so much for your patience. Thank you so much for taking the time to join me. We appreciate your continued interest in these webinars. And we have a new one every month. So again, keep an eye out for our December webinar, and we hope you'll join us again then. But on that note, thanks again so much for all your time, and until the next time, all the best.